Our second speaker is Stefan Pöhlmann. I think he is here. He is from George August University, Göttingen, from the Infection Biology Department, and he's also head of the Infection Biology Unit of the German Primate Center. And he's going to be talking about the SARS-CoV-2 infection. Please, Stefan.
Yeah, hello everybody. I would like to thank the organizers for the opportunity to speak. I would like to also ask for their pardon because part of what I'll present is not really so much immunology related. Bear with me, the second part will have a focus on immunology. Very briefly, my name is Stefan Pöhlmann. I'm working at the German Primate Center, and my long-standing research interest is to question how viruses enter target cells. And we've been looking at coronaviruses basically since SARS-CoV-1. We had, I might say so, a lot of experience. So when SARS-CoV-2 came around, we could make quick progress. And our initial findings indicated that the spike protein mediates entry. It needs two host cell factors for that, receptor ACE2 and the activating protease TMPRSS2. And finally, we found that this process can be blocked by antibodies. And basically, those findings kept us busy for the following 3 to 4 years of the pandemic. And before I present our findings, I would like to thank the people who did that, very particularly an excellent postdoc, a coronavirus aficionado and entry expert, Markus Hoffmann, as well as an extremely talented PhD student, now postdoc, Lu Sang, who joined the team. And of course, big thanks to every team member. What is the idea for today? At the beginning, I'll have some focus on spike and the activating proteases, and then I'll switch to the antibody responses. And the first focus is, unfortunately, a long-term topic, the furin cleavage site. Very simply, very early on in the pandemic, we found that there's this unique furin cleavage site present in the spike SARS-CoV-2, absent in the spikes of all related coronaviruses. And we were thrilled to find that that cleavage site was essential for lung cell infection. What we didn't know was the things to come on a political level, because as you know, now the furin cleavage site is considered as a potential marker for SARS-CoV-2 being generated or have been generated in the laboratory. However, science would say that the first cases cluster with the Wuhan fish mark, and also early variants are present there. And by the way, not directly, but distantly related viruses, distantly related SARS-CoV-2 viruses, they have multi-basic cleavage sites. So what did the field learn in the meantime regarding the multi-basic cleavage site? High impact studies relatively early on would indicate very simply that an intact furin site is needed for full virulence and is also needed for transmission. Meaning the virus, also none of the other viruses in nature did it, picked up this motive to become highly virulent and transmissible, or might have obtained the motive in the laboratory. And our data would actually kind of fit into this approach, because we could show that if we take very diverse spikes from SARS-CoV-2 related viruses from bats, we equip them with a multi-basic cleavage site. We see, let's see if I can point. Yes, I can. We see not a big effect, if any, in kidney cells. However, the spikes that have a multi-basic cleavage site now, and only in the presence of the multi-basic cleavage site, efficiently enter human lung cells. However, a recent preprint shows a slightly different picture. Basically, this study indicates that yes, the intact furin cleavage site is needed for full pathogenesis. is needed for efficient replication in the lung as expected. However, somewhat surprising, the multi-basic cleavage site does not need to be intact for high level of virus in the nose and for appreciable transmission of that virus. And that changes things a little bit because it would say that there has been the chance for precursor viruses not having a multi-basic cleavage site to enter the human population, and then they might have optimized that site on the way. Well, pre-switch, SARS-CoV-2 is not the only interesting clinically relevant coronavirus. There's the pre-pandemic MERS-CoV-2 that is transmitted from camels to humans, and so far, fortunately, didn't learn how to be efficiently transmitted between humans, but that might change in the future. And we thought, well, maybe the first step towards this unhappy change could be an optimization of the minimal furin cleavage site present in MERS-CoV-2. And indeed, when looking at patient sequences, we identified some with additional lysine residues at the cleavage site. And we found that this change markedly reduced cleavability, but to our surprise, it did not increase lung cell entry. Further, if we manipulated the spike of MERS-CoV in a way that we inserted multi-basic cleavage sites known from other viruses, including SARS-CoV-2, we were not able to increase lung cell entry of MERS-CoV, suggesting that it's not always the same mechanism that makes animal coronaviruses highly infectious and transmissible for humans. So very briefly, furin cleavage site is required for virulence, but is apparently dispensable for transmission. MERS-CoV can optimize its cleavage site, but this might be a long road, and just exchanging it against furin consensus motif is at least not sufficient. Let me stick also for this part to the protease before I come to the antibodies. This was also an initial important finding. Hydroxychloroquine had been postulated as the world savior against COVID-19. However, it didn't really work out because it blocks a surrogate pathway that is depending on cathepsin L, but it doesn't block the major pathway for SARS-CoV-2 entry into cells that is dependent on the protease temperase 2. And of course, this also got us interested. Clinical studies were run, and initially results were extremely disappointing. Nothing really did work, but also everything had in common that compounds to be tested were applied IV. Now, a simple change, if the same compounds were applied topically, meaning directly added to the respiratory tract, then potent antiviral activity was observed in animal models. And this got us excited enough to take it one step further and look at non-human primates. And also, variability is slightly higher than I would like. It's still of note that if the same inhibitor is applied via intravenous inoculation, there's really no effect of viral load. If the same inhibitor, however, is given as spray, then you have very potent antiviral activity. And in the meantime, we know that this works in humans. R-proteinin, similar protease inhibitor, has been nebulized, has been applied within a clinical trial to humans, and has worked just fine. Increases patient discharge from the hospital, less oxygen therapy needed. And therefore, I maintain that one of those much sought after broad spectrum antivirals that we need to be prepared for the next pandemic should be a temperase 2 inhibitor. Now we leave virology and focus a bit more on immunology, talking about antibodies and variants. A bit embarrassed to say we studied them extensively, but the major finding over the last years didn't really change so much. With the dominance of the Omicron variants, we have viruses that are highly neutralization resistant due to extensively mutated spike proteins, but still maintain the ability to efficiently spread in the upper respiratory tract, thereby ensuring transmission. What I find interesting is that this high transmissibility and antibody evasion apparently came with a price, because, as I just said, in the upper respiratory tract, well, yeah, they do find those variants, whereas in the lower respiratory tract, they do not have a problem with infecting lung cells. And this can be correlated with a change in protease preference. Thus, the early variants, sorry, that was one too much, the early variants all depend on TMPRSS2 unto delta, whereas Omicron chooses the surrogate pathway that is dependent on cathepsin. And of course, this raised the questions whether all Omicron variants do work the same way. And since I posed the question, the answer is no, that is not the case. There are interesting outliers. For instance, BA5, that has learned to enter lung cells with high efficiency, very similar to that to the wild-type virus. This entry is not due to altered protease choice, but works in animals and results in pathogenesis. Similarly, when looking at BA286, the precursor of the JN1 virus that was globally successful, again, we see that this virus has learned to infect lung cells with high efficiency, and in this case, efficient lung cell entry can be correlated with usage of this ancestral TMPRSS2 dependent pathway. So the point is, currently circulating Omicron subvariants on occasion can bring back interesting biological traits that we only know from earlier variants and that are associated with pathogenicity. Now, very briefly, how about the latest viruses? Here we have KP3, KP3.11 that have been circulating in the last couple of months. See no big difference in ACE binding. You see no big difference in entry, but you see that still the new virus, KP3.11, is doing significantly better in lung cells as compared to JN1. This is spike independent, and the underlying reason remains to be explored. So very briefly, some of the Omicron subvariants can relearn how to efficiently enter lung cells, and this is a trait associated with pathogenesis. Now, final point, sticking to the antibodies and focusing a little bit more on the animals or bacuviruses as compared to SARS-CoV-2. What do we have, and why is it interesting to look at antibody-mediated neutralization of those viruses? It is interesting because they show different entry phenotypes. Some strictly depend on ACE2 for entry. Others strictly do not need ACE2 for entry, but need trypsin activating protease for entry. Then the next group can do both. They can use ACE2, but they can also use the trypsin-dependent entry pathway. And then finally, there are some that just don't enter. Why is that important? It becomes interesting when we look at antibody-mediated neutralization. Some of those spikes just act normal. They are neutralized in a concentration-dependent manner. However, some within the RBD clade 1A spikes, which can choose between entry pathway 1 and entry pathway 2, here you get findings like in the presence of trypsin, neutralization is gone, indicating that this new pathway can protect from antibody-mediated neutralization. Now, how does this work in the context of vaccination? Here the simple message is, and this is actually a very good message regarding public health, if you are vaccinated sufficient times, ideally 3 to 4 times, then you'll have antibodies that appreciably neutralize all the spikes that we have tested. So you should have some level of protection against those potentially emerging viruses. But how about the ACE2 independent, trypsin-dependent entry pathway? Here it gets interesting. the spikes that are unable to use this pathway are neutralized very comparably in the presence and absence of trypsin. However, those spikes that can switch to the trypsin-dependent pathway, they become considerably less neutralization sensitive when doing so. In short, this study indicates that some sarbecoviruses can choose a trypsin-dependent, ACE2-independent entry pathway, and this pathway protects them from antibody-mediated neutralization. And this brings me to an end, which gives me the chance to thank again the people who did the work, particularly Markus, Lou, and Perna, but also our friends at MAH, Georg Behrens at Insel Riems, Martin Beer, Frank Kirchhoff at Ulm University, Christian Trosten here in Berlin, Stefan Pöhlmann, and Hans-Martin Jeck, and of course you for your attention.
Thank you so much for the beautiful talk. Any question or comments? Okay.
Nice insight into the details, how the virus gets into the cell, the ACE2 dependency, and also the trypsin aspect. My question to your trypsin data is, there are antibodies binding to the spike at the S1, S2 RBD domain, as we know. And you showed that the trypsin pathway it's neutralizing, or it's antagonizing the neutralizing capacity.
Yes.
Is that due to the inability of the other antibodies, for example, the S2 specific antibodies, to interfere with entry? Or is that something that just is so addicted to the RBD and the direct contact between the target cell and the virus that this sort of is the major effect by the trypsin is really RBD dependent? Why are the other antibodies out of the picture, or don't you have them in your system? Because I think the S2 antibodies are underestimated in their relevance. They might not be relevant for entry, but relevant for general immunity. But in your system, I'm not sure whether they could have an impact or not.
Thank you. That is an interesting question. I'm not sure we have sufficient data to address this. We made sure that the trypsin doesn't exert any unwanted effects like degrading the antibodies, which would be a major concern. So this has been controlled for, but the aspects that you suggest, we didn't, also because we only examined a very limited amount of monoclonal antibodies, but we should continue that. Thank you.
Yes, Andreas, have a question.
I have a question myself. So this shift from dependency on different enzymes, trypsin, catepsin, how does that actually impact on the further infectious potential in the body once the virus has, so to speak, passed the lung epithelium? Which cells would be are targeted actually by the early and late variants? In a different, is there any difference, or are other somatic cells like granule cells, neuronal cells, all actually susceptible to both?
I would try or like to try to answer this question in the context of the animal sarbecoviruses. For them, it makes sense because it's known, or at least very much suspected, that the GI tract is their major target. Trypsin is expressed at high levels in the GI tract, and most likely supports viral spread in this organ. And then the challenge is going to be, from my perspective, to make it to the respiratory tract, to acquire high transmissibility, and then this has to be accompanied by a choice in protease. But not sure I completely understood, but so far we did not test whether any of the circulating variants can counterintuitively but positively use that trypsin trypsin-dependent ACE2 dependent pathway as well. That would be very exciting, and we will be doing this. But so far we don't have data.
I will ask you that regarding to your findings, how this knowledge inform the development of antiviral strategies in general. Can you make any comment?
Well, as I indicated, with good conscience, I'm pretty bullish. I'm pretty convinced that inhibitors of activating proteases, specifically TMPRSS2, merit further exploration, and that is something that we'll be doing as well. The brief summary is we now know they are used, different viruses, not only coronaviruses, also influenza and others depend on the enzyme. We know that the enzyme is dispensable for development and homeostasis, it can be targeted. We have potent inhibitors, and initial trials indicate that if applied the correct way, directly the respiratory tract, those inhibitors work. And I think they should be in our plans for the next pandemic.
Along the same line here, Stefan, very nice talk. Does a TMPRSS2 inhibitor still work against Omicron because it uses the other proteases?
Yeah, we did this experiment, or more precisely, the group of Luca Cicin-Shain at Braunschweig HZI did that jointly with us. And it's very important to know that also there is a minor difference in TMPRSS2 dependence between early variants and Omicron. Omicron remains TMPRSS2 dependent.
Okay, there is no more question, I think. Thank you very much again.
Thank you.
Thank you.